Assalamu alaikum. My name is Tahir Ahmad and this is my video on the COVID-19 testing. An introduction. COVID-19, also known as coronavirus, is an infectious disease that affects the respiratory system and is caused by a virus called SARS-CoV-2. In the start of the pandemic, testing was not available and it was difficult to find out if a person had COVID-19 or not, so they went with checking for temperature. They assumed that if the temperature was high, which means they had a fever, they could possibly have the virus. But recently, a new way of finding out if the COVID-19 virus exists has been found, and it is called RT-PCR. What is RT-PCR and how does it work? Basically, RT-PCR is a method of finding out if the virus is present or not. First, we need to know that because the virus exists in the respiratory system, it begins with a swab of the patient's nose or the throat. Saliva and mucus from the patient's lungs can also be used. If the person has COVID-19, the swab will include a mixture of human cells, virus particles, and other microbes. After that, we use an enzyme called proteinase K. Proteinase K digests all the unwanted proteins and other microbes until the only thing that is left is the nucleic acid of the virus. Nucleic acid is a collective term for DNA and RNA. DNA is the genetic material in our body and RNA is similar to DNA. DNA and RNA are very similar to each other, but the only main difference is that DNA is double-stranded, this is one strand and another strand, while RNA is single-stranded. And another difference is that their bases, which are these lines here, these are called the bases. Out of these four here and four here, three of them are the same. Cytosine, adenine, and guanine are the same. The only difference is here, instead of thymine, there is uracil in RNA. So DNA has thymine, RNA has uracil. The virus that causes COVID-19, SARS-CoV-2, has RNA as its genetic material instead of DNA. For us to be able to check if the viral nucleic acid is present in the patient's sample, we need to change the RNA to DNA, and we need to create more of that sample of nucleic acid. So we put the RNA sample into a PCR system, which stands for Polymerase Chain Reaction System. The reason we need to change RNA to DNA is because the PCR system cannot replicate RNA, but it can replicate DNA. So now we will go on, I'll show you a video about this. So as you can see, there is not enough viral RNA to detect directly in the patient sample, so we use RT-PCR. This will amplify a gene called the N gene. The N gene is what we use to find out if the virus is present or not in the sample. So first we will take this RNA and we need to turn this into a DNA. First we use a DNA primer. A DNA primer is a short single strand of DNA that will let an enzyme know where to start and create one strand of a DNA. So this DNA primer will attach to this N gene. Once it does, it will join as so, and then an enzyme called reverse transcriptase will continue on and create the rest of this single-stranded DNA. Now that this is done, we have one part which is DNA and one part which is RNA. Remember, as I told before, that the only difference between DNA and RNA is DNA, there's two differences actually. One of them being the bases. DNA has a base of thymine, whereas RNA has uracil. So as you can see that both of these have blue, both of RNA and DNA has green, and both of them have yellow. The only difference is RNA has orange and DNA has red. So from this we will know that this orange is the uracil and the red here is the thymine. So this is how we know that one strand is DNA and the other one is an RNA. This DNA that we just formed, this strand, is called the complementary DNA or cDNA. But now we need to remove this RNA to make this whole thing DNA. So the RNA will be removed and then another DNA primer will attach to the complementary DNA, and then another enzyme called Taq DNA will create the rest of the DNA strand, making this one whole DNA strand, which is the SARS-CoV-2 and gene. But this time it's not an RNA, it's the DNA version of it. So now we need to amplify this. So what will happen is, first of all, they will separate, and then the primers will join, and then the Taq DNA polymerase will extend or synthesize a new strand of DNA, and then this will keep going on. And on. So, after 30 cycles of PCR, billions of copies of the viral cDNA are formed. Generally, you would need 30 to 45 cycles of PCR to detect the virus. After that, we use an object called a fluorescent probe to show us how many viruses are present in the sample. Fluorescence, as you might know, stands for light. So based on the light emitted, we will know how much viruses are present. As you can see in this graph, the amount of fluorescence is here, and the PCR cycle is here. Generally, now we can see that there's an increase after 30 PCR cycles, and after 45 cycles we can see a clear difference. So after 45 cycles, the blue or purple shows 200 viruses present, the orange shows 20 viruses present, yellow shows 2 viruses present, and then the green shows no viruses present, which is a negative result. That means the person does not have COVID-19, whereas these three show the severity or the amount of viruses present. Jazakallah. Assalamu alaikum.